And I want to start also saying that this is a long-term collaboration between my lab at MRC Harwell and Lizzie Fisher's and Linda Greensmith's lab at UCL Institute of Neurology. So I guess this slide is a bit of a self-explanation as to why point mutations are critical to try to understand TDP43 and fast proteinopathies particularly. But Bettina did such a great job on selling the point mutations that I probably skip a little bit. But just to say that we are not using CRISPR-Cas9 until now. Now we are moving towards that. So what we have been using is ENU mutagenesis, and ENU is a mutagenic agent that mainly produces random point mutations throughout the genome. So with ENU mutagenesis, it is the mutations that choose you. It's not you choosing the mutations. However, they are point mutations, and they have the clear advantage of being expressed at the right time, at the right place, and at potentially at the right levels, although that's not necessarily always true. Particularly for TDP43 and fast proteinopathies, I think point mutations are critical. And why is because they first allow to potentially disentangle loss of function phenotypes that perhaps transgenics are more challenging, but also because FAST and TDP43 particularly seem to be dose dependent. So toxicity, either wild type or mutant TDP43 overexpression seems to be toxic. And I'm not throwing a stone at transgenic mice, and I'm sure Kevin will show us amazing work in the next talk. But it's just that we are working towards a different problem potentially, in the sense, not a different problem, but having a different angle. And clearly all models are absolutely, to generate a wide variety of models is really critical if we try to understand something as complex as ALS FTD. So just very quickly, how we use the ENU mutagenesis in a reverse genetic fashion is because we at MRC Harwell, also in Germany and also in Japan, what we had over the years is to mutagenize thousands of mice with ENU and then cross them with wild-type females, and then get DNA and matching sperm from those F1 individuals that carry, on average, one mutation every 1.5 megabases. And we can screen that DNA for mutations in particular genes, autosomal mutations. So what we do is that we get and screen the DNA, and for particular genes, we can potentially do it with RNA-seq now, but probably CRISPR-Cas9 technology is superseding that now. And we've done that with a number of ALS genes, and we get the sperm, the matching sperm, and we can use it for IVF to generate the mutant mice. So we've done that for TDP43 using our MRC Harwell archive and also the Japanese archive. And I should point here as well that we have allelic series for other ALS genes, including SOD1, including VCP, including FAS. And we are, as I said, moving towards the CRISPR-Cas9 that we have already established in our institute, which is absolutely a great technology. So we found point mutations in all the major domains of TDP43. This is a really long-term issue. We were discussing before the aging in the mouse. An old mouse lives to 2.5 years. That's a pretty old one. So we really age the mice towards finding age-related phenotypes. So this is a very long-term process. So I'm going to focus on these 3 mutations today. And all the others, for these ones we had mice, we don't have them anymore, but for the others, either we do or in the Japanese archive, the sperm is available to generate the mice if you are potentially interested. So because we had so many mutations and we didn't know which ones will be potentially interested to work on, we decided to throw them to a functional assay. And what we did, Professor Baral and Dr. Burati talked this morning about the add-back assay, the CFTR exon 9. So what we did was to come here That was the work of a PhD student, Tom Ricketts, and throw all the mutations that we had to this add-back assay, where we siRNA the human TDP43 in HeLa cells and add back the mouse, which were resistant to the siRNA. And what we found was a potential outlier here on the F2101 mutation, F2101. So what was interesting about the F2101 mutation is that it is on the second RNA binding domain. And we thought that perhaps that deficit, which was somewhere in between the total knockdown and the wild type, was due to the efficient RNA binding. So indeed it was, and we did MSAs with recombinant TDP43 wild type or F2101 using UG repeats or also using the clip sequence here. And we found that indeed the F2101 mutation greatly diminished the RNA binding capacity of TDP43. So here we had a model where we can study the in vivo the function of RNA binding in TDP43, and if that can lead to disease-relevant phenotypes. So we have characterized, as I said, these three mice so far. So starting with the stop mutation, this was before the knockouts were published, and as with the knockouts, the homozygote stop mutations are null, are not viable, and unlike the zebrafish, I will say that in the mouse, the knockouts implant but don't survive very early. So at E6.5, there are no knockouts, as well as no stop mutant homozygotes. In the heterozygotes, the protein levels of TDP43 are the same as in the wild type, and that is due to the autoregulation process, as Professor Baral talked so nicely today. However, although the general protein levels of TDP43 in the brain and spinal cord are the same, we do, and also with the knockout, heterozygote knockout, there are some phenotypes on those mice, and we see some heel limb clasping, as Dr. Igas was saying on his mice, and also some body tone deficits. Moving towards the F2101, they are not viable. However, unlike the knockouts, they survive, and depending on the genetic background, we can take them towards E18.5. Bettina did a great job at analyzing the zebrafish knockouts. We are not there yet with these mice. However, they are also developmentally delayed. So in general, they are around 2 days delayed. It seems that organogenesis is fine. Everything is there. It's just that it is potentially delayed. You can easily see a spinal edema here, but that actually resolves. And by E18.5, I don't have the picture, you have a normal, if you want, E16 or 17.5 embryo. So this resolves out. So moving towards the M323K mutation, why we were interested in this one is because it's at the right place, although there are no pathogenic mutations described on this residue, but it's in the prion-like domain, so we wanted to study them as well. So on a black 6 background, keeping back crossing to black 6, the homozygote mutants are not viable. However, if we do the trick of getting some genetic variation by crossing to DBA, we actually can rescue the homozygote phenotype, and that has allowed us to study the homozygote phenotype in the adults. So quite interestingly, early on, we realized that, again, Professor Baral and Burati were talking about the CFTR minigene. What we found was that the F2101 mutation had a loss of function signature for the CFTR minigene, but very interestingly, the MC23K had the opposite. So it behaved as a gain of normal function. So somehow the MC23K mutation affects the splicing function of TDP43 by making it more efficient. And this is also true for different endogenous genes, including sortilin, but also poly-DP3. If we compare this in a genome-wide manner, using embryo heads to compare between the different genotypes, what we see is that we see a correlation, a negative correlation, where exons that are upregulated in the F21 are downregulated in the MC23K. So we found this quite interesting, although doesn't necessarily relate to the human condition. Saying that, there are some reports, the last one on the siRNA PNAS paper, the siRNA knockdown mice, where they show from spinal cord of patients that there were a mix of exons that were having the signature of a loss of function, of TDP43 mediating splicing. Some had a loss of function signature, but also at least one had a gain of function signature. So that's interesting, and we are definitely following how mechanistically how this mutation can actually affect the splicing and being more efficient. So one very crude way that we try that here as well with Emmanuel and Tito was to do a semi-quantitative EMSA assay with recombinant protein, but we couldn't really find any differences, meaning that at least with this assay, the MC23K mutation doesn't really bind RNA better than the wild type, which doesn't mean that is not going to be the same in vivo. At least, you know, in this overexpression system, it doesn't. Another possible explanation is that the protein levels are different. And however, the protein levels for MC23K, but also interestingly for the F21, they are not different, the TDP43 protein levels. And that's interesting because for the F21 mutation, because of the autoregulation process, we will expect to have higher protein levels because this TDP43 F21I mutant is not able to bind as well the 3' UTR of its own RNA, and we have shown that in vitro. However, that doesn't lead to difference in expression levels of the protein, either nuclear or cytoplasmic. And the same is with the MC23K, and we are a bit puzzled about this at the moment. So we, in collaboration, I should stress that this is collaborating work with Lizzie Fisher and Pietro Fratta, and some of you were probably at the NCLS meeting last week, and Pietro presented this data as well. So one really strange result that we had with the RNA-seq is in terms of the splicing levels. The F21 mutation is like a knockdown. If you compare the data to Don Cleveland's data, you see that the signature in terms of the splicing of the cassette exons going in and out is exactly the same between the knockdown and the F21I mutation. However, we heard yesterday about these long intron genes, and which from Don Cleveland paper, you see that they are enriched. These are downregulated here. So these long intron genes are enrichedly, and they are downregulated with a knockdown. However, with our TDP43 F21 heterozygous and homozygous, they are actually upregulated. So I should stress here that this is RNA-seq data, and we haven't really validated this yet on RNA-seq, on qPCR, so we really need to do that. It's very preliminary. But it looks like the effect on the long intron genes is opposite from the knockdown with the F21 mutant, and that's really puzzling. We don't really know how that might be. So just to finish, to be able to have the mice really allow us to characterize and see if the aging process and not also development have what phenotypes do these mice develop. And to our despair, I should say, and that links also with Bettina, that by mutating the endogenous genes, you potentially need second hits. The F21 mice, the only phenotype we can find is that the females weight less than the wild type. This is on the heterozygote ones. And remember that these heterozygote mutants have a signature of a knockdown. It's dose dependent, but they do have really amazing changes at the molecular level. However, the mice are fine, and we have looked quite hard to try to find some phenotypes. This effect on the weight is also seen on the homozygote MC23K mutants. Although this is on males, I should say, before they are. So we are able to see some sexual dimorphism here. And finally, just data from a couple of weeks ago, it seems that the MC23K mutation can affect the cognition of the mice. So this is in a fear conditioning paradigm. The MC23K homozygote mutants, and to a lesser extent the heterozygotes, can actually affect the freezing time on the queue, but not in the context. So this is not significant. So to conclude, missense mutations in TDP43 can selectively affect TDP43 functions and give us a novel model systems to try to dissect which functions of TDP43 may or may not be related to disease. However, the changes are subtle and potentially need a second hit. And again, we are thinking on using genome editing technologies to provide those second hits, as well as crossing different mice and so on. In terms of the F2101 mutation, first, a mutation in the second RNA binding domain can clearly affect the RNA binding, and that's also novel, but also give us a model system to work on, because the embryos actually survive to 18.5, and we can make embryonic motor neurons, cortical neurons, and MEFs that we are continue to study and have already led us to quite, I think, interesting biology that I'll be talking in the next meeting, hopefully. And then a mutation within the glycine-rich domain, although it's not a pathogenic mutation, but we are making them now at the moment with CRISPR-Cas9 technology, can actually affect the splicing function. And after Emmanuel's talk this morning, we are definitely going to be looking at potential differential interaction with HNRMP-A2. So I should say that we did the Far Western, the GST overlay, and there were no qualitative differences. But we're definitely going to try for the IPs. And just to finish, this is a very collaborative work between Lizzie's lab and my lab at Harwell, and it's mainly the work of very talented clinician Pietro Fratta and Tom Ricketts and Hugo Oliveira at my lab. Thanks a lot.